Source: Indiana University School of Medicine research published in Alzheimer's and Dementia, the Journal of the Alzheimer's Association. If you like my videos, subscribe my channel and press the bell icon to turn notifications on so you keep getting all the major advancements in science and technology. And don't forget to like and share this video with your friends and colleagues. Comments are welcome and open for discussion. For decades, Alzheimer's disease has remained one of the most devastating and mysterious disorders in medicine. Millions of people worldwide slowly lose their memories, cognitive abilities, and eventually their independence as the disease progressively damages the brain. Scientists have long known that Alzheimer's is associated with the buildup of amyloid plaques and abnormal tau proteins, but understanding exactly what drives these destructive processes and how to stop them has proven extraordinarily difficult. Now, researchers at the Indiana University School of Medicine may have uncovered an entirely new biological trigger hidden deep within brain cells themselves. In a breakthrough that could reshape future Alzheimer's treatment strategies, scientists identified a specific enzyme known as IDLE that appears to play a critical role in the development and progression of the disease. Even more remarkably, when researchers removed this enzyme in laboratory experiments, the brain showed dramatic improvements, including reduced amyloid plaque accumulation and stronger cellular processes linked to resilience and communication between neurons. What makes this discovery especially important is that most current Alzheimer's treatments focus almost entirely on removing amyloid plaques after they have already accumulated in the brain. Recently approved drugs such as lecanemab and donanemab are designed to clear these plaques and slow the progression of cognitive decline. While these therapies represent major advances, they do not fully stop the disease, nor do they repair the underlying damage already occurring inside neural networks. The new research suggests that targeting IDLE may offer a fundamentally different strategy, one that not only reduces harmful plaque buildup, but may also strengthen the brain's ability to resist degeneration itself. The enzyme at the center of the study has a surprisingly unusual name: IDLE, short for inducible degrader of the LDL receptor. Scientists originally became interested in this enzyme because of its role in regulating lipid metabolism, the complex system by which cells process fats and cholesterol. The brain relies heavily on lipid regulation because neurons require enormous amounts of energy and structural support to maintain healthy communication pathways. Disruptions in these processes have increasingly been linked to neurodegenerative diseases, including Alzheimer's. Researchers suspected that IDLE might influence some of the molecular systems connected to plaque formation, inflammation, and neuronal survival, but the full extent of its role remained unknown. To investigate the enzyme's function, the Indiana University team engineered multiple experimental models in which the IDLE gene was selectively removed from different types of brain cells. Initially, scientists believed the most significant effects would appear in microglia, the brain's specialized immune cells responsible for clearing waste and damaged material. Microglia are major producers of IDLE and are deeply involved in inflammatory responses associated with Alzheimer's disease. Surprisingly, however, the most dramatic improvements emerged not when the enzyme was removed from immune cells, but when it was deleted directly from neurons themselves. Once IDLE was removed from neurons, amyloid plaque accumulation dropped significantly. But the findings became even more intriguing when researchers examined other molecular changes inside the brain. They observed reduced levels of a polypoprotein E, commonly known as APO, a protein strongly associated with Alzheimer's risk. One specific form of this protein, APOE4, is considered the single greatest genetic risk factor for late-onset Alzheimer's disease. APO plays a major role in lipid transport and metabolism inside the brain, and abnormalities in its regulation are believed to contribute to plaque formation, inflammation, and neuronal dysfunction. The fact that neuronal IDLE deletion altered APO-related pathways suggests that the enzyme may sit at a crucial intersection connecting multiple disease mechanisms simultaneously. Even more importantly, the study revealed increases in receptors involved in maintaining neuronal communication and lipid balance after idle removal. These receptors are essential for preserving healthy signaling between neurons, allowing brain circuits to function efficiently despite ongoing stress or damage. Earlier research has shown that strengthening related pathways may help certain individuals remain cognitively resilient, even when significant amyloid plaque accumulation is already present. In other words, some brains appear capable of tolerating pathological changes far better than others. The new findings suggest that blocking idle could potentially enhance this resilience, allowing neurons to better withstand the disease process rather than simply slowing plaque formation alone. This distinction could be enormously important from a clinical perspective, because Alzheimer's is typically diagnosed only after substantial brain damage has already occurred. By the time symptoms become noticeable, plaques and other pathological changes may have been developing silently for decades. Therapies capable of both reducing toxic buildup and strengthening neuronal survival mechanisms could therefore provide much greater benefits than approaches focused solely on plaque removal. Researchers believe targeting neuronal idle may simultaneously reduce amyloid burden, improve lipid metabolism, preserve synaptic communication, and potentially enhance the brain's natural protective responses against degeneration. The implications extend beyond Alzheimer's itself. The study highlights how modern neuroscience is increasingly moving away from viewing neurodegenerative diseases as simple, single-cause disorders. Instead, scientists now recognize that conditions like Alzheimer's emerge from highly interconnected networks involving inflammation, metabolism, protein aggregation, immune responses, and cellular signaling. Enzymes such as idle may act as key regulatory nodes within these networks, meaning that modifying a single molecular target could influence multiple disease pathways at once. The research team is now focusing on the next major challenge: translating the discovery into real therapeutic compounds. Because enzymes possess well-defined molecular structures with active binding sites, they are often excellent drug targets. Scientists can potentially design highly specific molecules capable of attaching to the enzyme and blocking its activity with minimal side effects. Future studies will evaluate the safety of potential idle inhibitors, test their effectiveness in advanced preclinical models, and investigate whether suppressing the enzyme can also reduce tau pathology, another central feature of Alzheimer's disease that contributes heavily to cognitive decline. Perhaps most importantly, this discovery offers something Alzheimer's research desperately needs: a genuinely new direction. For many years, progress in the field has been slow and often disappointing, with numerous experimental therapies failing in clinical trials despite promising early results. The identification of idle as a potential master regulator opens an entirely different therapeutic avenue, one focused not only on removing damage, but on actively helping the brain defend itself against degeneration. Although substantial work remains before any new treatment reaches patients, the findings represent a powerful reminder that the brain still contains hidden biological mechanisms scientists are only beginning to understand. Deep inside neural cells, enzymes, like idle, may quietly shape the balance between degeneration and resilience. By uncovering and controlling these hidden systems, researchers may eventually move beyond simply slowing Alzheimer's disease and toward truly protecting the brain from its devastating effects altogether.